Okay, so I think we can start. Good morning, everyone. Thank you for attending my thesis defense, either in person or remotely. I would also like to thank the member of the jury who took the time to evaluate my work and who are here today. I represent my thesis work, supervised by Olivier Maury and Mathieu Langagne, which focuses on assessing the impact of climate change on global marine ecosystems. In my thesis, I studied the pelagic zone, which refers to the open ocean water column. This water column is organized vertically according to density, light, temperature, and nutrient gradients. We have the epipelagic, mesopelagic, bathypelagic, abyssopelagic, and hadopelagic zones. My work only considered two zones, the epipelagic zone between the surface and approximately 200 meters, and the mesopelagic zone between 200 and 1000 meter depth. Despite significant growth in research efforts, pelagic ecosystems remain relatively poorly understood compared to coastal and inland ecosystems, particularly deep pelagic ecosystems like the mesopelagic layer. The pelagic environment hosts a diversity of marine life. The first link in the food chain is phytoplankton. These microscopic organisms live in the surface waters where they carry out photosynthesis and form the basics of pelagic food webs. Then comes zooplankton. They comprise a wide diversity of organisms and exert significant control over larger organisms by acting as a key food source and recycler of organic matter. While plankton have limited locomotion, nectons are animals that can swim against currents. They occupy separate ecosystems based on their ability to move horizontally and vertically and tolerate different temperatures, oxygen concentrations, and light levels. So, plankton are considered as low trophic level organisms, while pelagic nectons are high trophic level organisms. It is these last organisms that I am studying. So, we have the environment and we have the organisms. But to get an ecosystem, they need to interact. And how do they interact? First, there is a temporal dimension to consider. Day and night, with light influencing organisms' behaviors. Epipelagic organisms inhabit the epipelagic layers, feeding with visual predation, so mainly during the day. Particulate organic matter includes detritus, fecal pellets, and animal carcasses. This marine snow sinks to the deeper layers of the ocean, where it is joined by deep zooplankton. They are a food source for mesopelagic organisms. However, organisms do not necessarily stay in one layer. Some mesopelagic organisms migrate to the surface at night to feed, hidden from predators by the darkness. And some epipelagic fish dive into deeper water during the day to feed on the abundant mesopelagic organisms. As you can see, organisms interact with each other directly or indirectly through complex prey-predator interactions, which determine how energy and biomass are transferred between high trophic level organisms and across trophic levels, a process known as trophic functioning. This trophic functioning arises from the spatial and temporal co-occurrence of organisms. They must be in the same place at the same time to interact. However, these interactions remain poorly studied, with limited consideration of the vertical structuring, despite the fact that it exerts a strong control over trophic functioning. So it is crucial to take into account the vertical dimension in order to accurately represent the trophic functioning of ecosystem. By using a 3D model, the aim of my thesis is therefore to improve our understanding of pelagic ecosystem functioning by identifying the key processes that govern trophic interactions across pelagic ecosystems. However, we need to consider the diversity of pelagic ecosystems. These ecosystems vary considerably depending on the time scale. We have observed the influence of days and night alternation. However, the seasons and other long-term variations may also affect the structure of ecosystems. Spatial distribution also plays a role. Ecosystems can differ greatly from one region to another. For example, cold waters host different species compared to tropical waters. But we have only just begun to understand the complex vertical and horizontal movement patterns of pelagic organisms and the factors that influence them. One of the aims of my thesis is therefore to identify the key physical and biogeochemical factors that structure pelagic ecosystems. To address these questions, models have gained increasing interest as a complement to observational studies. They aim to abstract and simplify reality in order to focus on key features. Marine ecosystem models have a relatively short history, beginning in the 70s. During the early stages of developing marine ecosystem models, the focus was primarily on environmental factors such as physics and biogeochemistry. Over the past few decades, our understanding of marine ecosystem functioning has increased significantly. At the same time, the growing number of environmental projects focusing on the sustainable use of marine resources has led to an increased demand for marine ecosystem models. This has required more complex and multi-component models that incorporate all ecosystem components from the dynamic of the abiotic environments to primary producers and top predators. These models are sometimes called end-to-end models. Cross-model comparisons can help quantifying uncertainties in model-based projections. For Earth system models, we have the CMIP, Coupled Model Intercomparison Project. And for higher trophic level, we have FISHMIP, Fisheries and Marine Ecosystem Model Intercomparison Project. FISHMIP aims to answer various questions about the future: fisheries and seafood supply, marine biodiversity, and functioning of marine ecosystems. This community gathers global and regional models, with some models representing ecosystems purely by body size or trophic level. Some are trait-based and size structure model, which mean they include different communities and groups using functional traits other than body size. And other are species-centered. During my thesis, I focused on one model in particular, the APECOSM model, which is part of FISHMIP. But I developed a new configuration to add more complexity to the ecosystem. So before using it for the next FISHMIP study or to analyze any organism behavior, I need to ensure that this new configuration of APECOSM is able to represent appropriate responses of different communities in comparison with observations. This is particularly important if I want to study climate change, with phenomena that have no equivalent in the past or present. With industrialization, human activities increased atmospheric concentrations of greenhouse gases, mainly resulting from the burning of fossil fuels. As a consequence, the atmospheric temperature increased abruptly over the past 50 years. But as oceans absorb a large part of excess atmospheric CO2 and heat, it results in multiple impacts on the oceans. As you may know, the sea surface ocean temperature increases. But temperature change impacts water density, leading to the expansion of stratified waters with less ocean layer mixing. Ocean currents are also altered, becoming stronger or weaker depending on the latitude. Biogeochemistry is not left behind, with the increased stratification and warm water leading to a decrease in global oxygen concentration, but also a decrease in nutrients in surface waters that are essential for primary production. As a consequence of the previous impacts cited just before, primary production also decreases. These changes in physics and biogeochemistry have already been observed to impact higher trophic levels. First, the metabolic rate disturbance, with, for example, increased metabolic rate with warmer waters. The average size of fish also changes, often shrinking, but not always. We also observe shifts in distribution areas and species migration, primarily through higher latitudes where the water is colder. Lastly, changes in primary production affect the entire food web, causing multiple disturbances from one region to another. However, all of these observations on the higher trophic level are species-specific, region-specific, and relate to the past and present. But what about the future? FishMIP has already evaluated the impact of climate change on marine animal biomass. Under a high-emission scenario, in red, total biomass is expected to decrease by nearly 20% by the end of the century. The distribution also changes, with less biomass at the equator and an extension toward the poles. But as all models are different, some with species, others with communities, or only one total biomass, it is necessary to aggregate data for comparison purposes. Consequently, these FishMIP studies on a global scale only look at the total animal biomass, which does not allow for an in-depth analysis of the mechanisms and processes at play. What about tuna responses compared to mesopelagic fish? Are they affected in the same way? And how might interactions between organisms evolve? All of these interrogations are addressed in my thesis under these three questions. Do the different pelagic communities respond in a homogeneous way? What processes are involved? How will ecosystem functioning evolve in response to potentially diverse community changes? So, as you may have heard in the questions, I will look at the results by community to overcome the limitations of total animal biomass. So we raised a lot of questions during this introduction. First, is the APECOSM model able to represent appropriate responses for each community in comparison with the observed phenomena? Which physical and biogeochemical factors structure marine ecosystems? What processes govern the trophic functioning of pelagic ecosystems? And under climate change, we have the three questions mentioned on the previous slide. In my thesis, we focused solely on ecosystem dynamics, which means that fisheries are not considered. Before answering these questions, I will first present the model and the configuration I used. I will then answer the first three questions by presenting my first chapter, which assesses the model and analyzes the structure and functioning of ecosystems on a global scale. This chapter relates to a paper published last year in Progress in Oceanography. Then I will introduce my second paper that answers these same questions, but this time on a regional scale. Published early this year in Progress in Oceanography 2, it aims to demonstrate the ability of APECOSM to represent contrasting ecosystems with one generic configuration. Due to the lack of time, I decided not to present my third chapter, which is about the influence of nitrogen fixation on marine ecosystems under climate change conditions. But this paper, hopefully, will soon be submitted. So let's jump straight to my first chapter, in which I analyze how marine ecosystem models respond to climate change. This paper, being written, aims to answer the last three questions. And of course, to conclude this presentation, a conclusion. So let's dive to the model presentation, APECOSM, for Apex Predators Ecosystem Model. APECOSM is a 3D model with a mechanistic approach. It is based on fundamental processes and theories, such as the dynamic energy budget, a theory that describes how energy is taken up and used by an organism for physiological needs, such as growth and reproduction. APECOSM is also a trait-based model. It means that rather than focusing on species, we group organisms with similar traits into communities. These traits determine their life history, such as feeding behavior, maximum size, or temperature preferences. The relative proportions of small and large species fluctuate dynamically within each community, altering its characteristics. The phenomenon of schooling is also considered and controlled trophic interactions. Depending on size and their distance to others, organisms will aggregate and become more or less detectable to predators. The model also includes the passive transfer by current, the active movement of organisms, and it also considers ecological processes such as opportunistic size-structured trophic interactions. The principle is to model low-level processes and see how the structures and mechanisms of ecosystems emerge from these laws. APECOSM is forced by a physical model, NEMO, and a biogeochemical model, PISCAS. Nemo gives us the current, temperature, the 3 dimension, and a 1 degree grid. Pisces gives us the light, the plankton, and the dioxygen. They interact with each other and enable APECOSM to simulate high trophic level communities. The previous configuration included 3 communities: the pelagic community, the mesopelagic migrants, and the mesopelagic residents. But epipelagic organisms have a strong ecological impact, while anchovies and tuna support important fisheries. To consider the diversity of responses of epipelagic organisms, we increase the complexity of this community by splitting it to 3, then 4 communities. So, the configuration I developed includes 6 high trophic level communities. We have the small coastal pelagic, small and medium epipelagic, and tropical tuna that live permanently near the surface. We then have the mesopelagic migrants that live at depth but feed at night near the surface. Mesopelagic feeding tuna live near the surface but feed during the day at depth. And we have the mesopelagic resident that inhabit permanently at depth. APECOSM communities are configured using various parameters. Finding the right parameters was the first stage in my thesis. I mainly worked on those for the small coastal pelagic community that are almost created from scratch. And I also worked a lot on the oxygen responses of communities. For this, I used the scientific literature and fishery data. So inside each community, we have the maximum size of species. For example, from 11 to 84 centimeters for small and medium epipelagic community. We also have large communities up to 2 meters or even 2.5 meters for mesopelagic feeding tuna. The depth position is determined by light ranges. Epipelagic communities are sensitive to strong light, which keep them close to the surface, while mesopelagic communities prefer darkness. Most communities are diurnal, meaning they feed during the day, while mesopelagic migrants feed at night. The diet is partly controlled by the size ratio between predator and prey, which is the same for all communities, except for small coastal pelagic, which feed on organisms much smaller than themselves, plankton in particular. Temperature preference regulates mainly epipelagic communities. Tropical tunas live in warm waters, small coastal pelagics in cold waters, and the 2 other communities have a wide temperature range, being considered temperate. The communities are also sensitive to oxygen. Epipelagic communities have high oxygen requirements, while mesopelagic communities are more tolerant to low oxygen concentrations. To force the model, we use drivers under a chosen scenario. For my first 2 chapters, I'm interested in the historic and present day. So I will initialize the model with a spin-up that only considers natural variability. And I will then run my simulation from 1958 to 2022 that include climate change over this period. Now that we have all of this information, I will present my first chapter that aims to assess the new configuration and study the structure and trophic functioning of marine ecosystem on a global scale. Here you see the biomass distribution from a model based on acoustic data. You can see that biomass is stronger in upwelling, coastal to the continent, and when the water is rich in nutrients. On the other hand, the biomass is lower in gyres, where the water is poor in nutrients. On your right, you see the geographical distribution of all high trophic level communities in APECOSM. This spatial distribution of high trophic levels is first consistent with the distribution of primary production, but it also has similar patterns with the model based on acoustic data on the latitude shown. This demonstrates the consistency of the APECOSM results. But since the ocean is 3-dimensional, we can also observe spatial distribution vertically. In APECOSM, it is influenced by several factors: light sensitivity, temperature preferences, prey-predator relationships, and oxygen requirements. To parameterize the response to oxygen, I used acoustic data from several geographical regions collected during an expedition called Manaspina. We can thus observe a general pattern where oxygen is not limiting, the blue dots, and another pattern in oxygen minimum zone, the green dots. This graph shows the vertical distribution according to depth. On the left, the day, on the right, the night. We see significant biomass at depth during the day, and some of this biomass rises to the surface at night. It's our mesopelagic migrants. In oxygen minimum zone, all biomass at depth disappear completely at night. This means that mesopelagic residents cannot inhabit these waters. But mesopelagic migrants can, rising 200 meters higher than the general pattern. After several iterations, I managed to find a similar vertical distribution in APECOSM. As you can see here, the model results, shown as dotted lines, satisfactorily capture the distribution found in the observation, the solid lines. This is true not only for the general pattern, but also for the low oxygen pattern. Another ecological characteristics that enable the assessment of the APECOSM model is the size spectrum. In the size spectrum, aquatic biomass is evenly distributed between size classes based on the logarithm of body mass. Basically, the larger an organism is, the less abundant it is. And this weight-abundant relationship forms a straight line with a slope of -1 on a log-log scale. Here are the size spectra in APECOSM on a global scale. The y-axis represents the biomass density and the x-axis the weight, both in log scale. We have the plankton from diatom to macrozooplankton. It's our input data. And we have the sum of our 6 communities from APECOSM. Thanks to the biological processes implemented, I managed to accurately reproduce this law with the sum of the communities aligning with the plankton. The most abundant communities are the mesopelagic ones, which are known to dominate the global marine biomass. We then have the epipelagic communities, and finally, we have the tuna communities that are large organisms and consequently less abundant. But if I parameterize the community to obtain this slope on a global scale, it does not mean that it is accurate for a specific region. So let's zoom in some of them. For the Indian Ocean, a comparison was made with the IOTC tuna catch data, IOTC for Indian Ocean Tuna Commission. Careful, here we compare only the slope, not the abundance, as APECOSM represents organisms' biomass, while IOTC represents fishery catches. So you see here the curve follows roughly the same slope, but with peak catches when tuna tend to school more, making them more catchable by seiners. The first term corresponds to their intermediate size, where they school a lot, and the second one to the reproduction stage, where they also school a lot. In the Atlantic Ocean, we have recovered data from the Triatlas project. It is more than 20 years of scientific expeditions with sampling records on phytoplankton, zooplankton, and mesopelagic organisms. If we compare the same areas, we find not only the same slope, but also the same order of magnitude in terms of abundance. So you can see here with the dots that represents Triatlas data and the solid line with the APECOSM outputs. And the results are quite similar. And now finally, let's look at how ecosystem functioning is determined by trophic interactions. So here, I recall that APECOSM is a mechanistic model, which means we do not control who eats whom. These results emerge directly from the structure of ecosystem, especially the spatial distribution of predators and prey, and the size range to which predators have access. Here we have the tropical tuna. The y-axis is the functional response that measures the degree of satiety. At 1, they are satiated, and at 0, they are starving. We have the small tuna on the left and the large tuna on the right. When tuna are small, they tend to eat on small prey, like mesozooplankton. But as they grow, they incorporate larger prey, like macrozooplankton, small fish, and even tuna. Mesopelagic feeding tuna have a similar behavior, especially when they are young, but as they grow, they start to dive deeper in the ocean to prey on mesopelagic organisms. That's why you can see that they incorporate mesopelagic migrants and mesopelagic residents in their diet when they are large organisms. Based on these emerging diets and considering the greater abundance of small organisms, we obtain the following trophic fluxes. The trophic fluxes are energy fluxes transferred from a prey to a predator group. Here, for example, we see that mesopelagic residents rely highly on particulate organic matter coming from the upper layer. Overall, this shows the multitude of interaction between each organism and highlight the interdependence between the epipelagic and mesopelagic zones. So in this chapter, we demonstrated the ability of the model to represent observations on a global scale using generic individual processes that enables the emergence of 3D ecosystem structures and functions. We saw that marine animals are impacted by physical and biogeochemical processes that influence horizontal and vertical distribution, abundance, size distribution, and trophic interactions. This chapter also demonstrates the strong interdependence of community through the water column. Now, let's study the model in a more regional scale. To do this, I will draw on my second chapter. Here, I present the APECOSM model's ability to represent contrasting ecosystems from a single configuration. The Pacific Ocean is the largest on the planet and exhibits high diversity. As seen before, the gyre, coastal biomass, the blue patches, while the upwelling regions, coastal to the continent, have strong biomass. Today, I want to focus on two key regions for fisheries, the Humboldt Current System and the Pacific Whirlpool. For global marine ecosystem model, it is a major challenge to represent such contrasting regional ecosystem structure and functions with generic design and rules. Capturing the key processes and their interactions at different temporal and spatial scales without excessive parametrization and computational cost requires a careful modeling strategy. This is what the APECOSM model is aiming for. So let me introduce you to our two regions. The Humboldt Current System, a highly productive upwelling system that supports important fisheries of small pelagic fish like anchovies, but also sardines. It is characterized by nutrient-rich water on the surface due to the upwelling, enabling high-intensity phytoplankton production. And we have an oxygen minimum zone at depth. On the other hand, we have the Pacific Whirlpool that supports a high diversity of marine life, including large top predators such as tuna, making it one of the world's most productive tuna fisheries. It is characterized by seamount presence, warm water accumulation from the trade winds, and stratification of the water, inducing low nutrients in the surface. To understand the characteristics of the ecosystem structure, let's visualize the size spectra of our two regions. Here we have the Humboldt size spectra, again with the plankton, our input data, and our 6 high trophic level communities from APECOSM. What we see is that small coastal pelagic fish in pink have high biomass, which is relevant with anchovy abundance in this region. About mesopelagic communities, we have more mesopelagic migrants than mesopelagic residents, which is the opposite of what is observed on a global scale. I will detail why later. Another interesting point is the strong seasonal variability, identifiable by the thin lines, monthly spectra, which indicates that biomass fluctuates considerably depending on the time of the year. In the Pacific Whirlpool, the size spectrum is very different. The total biomass is lower, although not that obvious on the log-log scale. But what is clear is that there is no cold water communities such as small coastal pelagic fish. In this region, tuna biomass is significant, which matches fishery data. And last but not least, the size spectra in this region are very stable, indicating little change in biomass all year long. Now, let's take a closer look at the Humboldt region. Here we have the biomass of small coastal pelagic along the transect. On the left, the north, on the right, the south. Below is the diatom concentration. Looking at these figures, we can see that the fish are partly influenced by diatom concentration. When diatom concentrations are low, the biomass of small coastal pelagic decreases too, which is characteristic of the bottom-up control. Looking at the trophic fluxes along the transect, we see that depending on the zone, the community can adapt their prey proportion to the available prey, from diatom in green to mesozooplankton in yellow. Not shown here, temperature also influences biomass at both extremities of the transect, seasonally too warm in the north and permanently too cold in the south. Mesopelagic residents, on the other hand, are greatly affected by the oxygen minimum zone. We can see their distribution in depth as a function of the latitude. So, represented in blue, they completely disappear at the core of the oxygen minimum zone. Mesopelagic migrants, in green, are more resilient as they rise to the surface at night, where waters are high in oxygen. Consequently, mesopelagic resident biomass in this region is lower than those of the migrants. In the Pacific Wampool, primary production comes from exported nutrients from the colton, as well as from small upwellings near seamounts and islands. Here, the response of tuna to the dynamic is clearly visible. We have their biomass distribution along the depth as a function of the longitude. To the right, large biomass is exported from the eastern region and enters the Wampool due to the currents and active movement. It makes the Wampool a hub region where all the trophic chain is advected from the east and is then exported northward and southward. In oligotrophic waters near Indonesia, primary production is deeper. Tuna also have a deeper distribution following their prey feeding on it. This region also exhibits seamounts, which impact the local community structure and behavior. Mesopelagic communities, the migrants in particular, rise to the seamounts at night where they tend to aggregate. As the day comes, they are trapped in the epipelagic zone, which increases the foraging opportunities of apex predators. This phenomenon is also observed on the real ocean. You can see the signature of seamounts present in the mesopelagic migrants' cooling probability. Now remember, in the model, feeding regimes are emerging, not controlled. These figures represent the trophic fluxes of tropical tuna and illustrate how they are incorporating mesopelagic communities into their diets, despite typically feeding on epipelagic prey on the rest of the transect. So to conclude this part, APECOSM is able to reproduce the main regional patterns, demonstrating its ability to represent a diversity of ecosystem responses to contrasting environmental regimes from first principle processes at the individual level. It also demonstrates the plasticity of pelagic ecosystems and their adaptability to different environmental conditions. Last but not least, it demonstrates that regional ecosystems are open systems that cannot be considered in isolation. The passive transport of biomass and active movement from source regions, such as the Humboldt, to hub regions, such as the Wampool, are functionally essential. I didn't present it today, but I also studied the gyre in my paper that could be considered as an ecological sink where a local demographic deficit occurs. For the next chapter, I will present my last paper that is still being written. Here we aim to understand how ecosystem structure and function evolve under climate change. So we are using other forcing scenarios compared to previously. We have two simulation references. One with our factor without climate change, it's our control baseline, and one with our factor under climate change, which is the high emission scenario, the SSP 5.5. But to analyze the influence of each factor, I also ran sensitivity experiment by applying climate change forcing to one driver at a time, while keeping all other drivers under control conditions. I did this for plankton, here under climate change conditions, while light, oxygen, and temperature are not driven by climate change. Also did this for the temperature, the oxygen concentration, and the light penetration. So this simulation isolates the individual contributions of each driver to ecosystem responses. But about temperature, two elements control community responses: the thermal preference and the metabolism. So I checked the thermal preference, applying climate change only on this driver, and I did the opposite, only looking at metabolism. And because we love running simulations, I also ran metabolism responses on epipelagic communities independently of mesopelagic communities and vice versa. So I had 10 simulations to analyze. With these simulations, first I compared the simulation with all factors under climate change condition with our control baseline. So this is the evolution of the APECOSM community biomass from 1958 to 2087. The total biomass of the community decreases by almost 20%, which is similar to the fishmeal result presented in the introduction. But when we look at the different communities, the results are highly heterogeneous, with mesopelagic communities that are way more resilient than tuna communities. Mesopelagic migrant decreases by 40%, while mesopelagic feeding tuna drop at minus 71%. Their distributions have, however, similar patterns. Here we have the distribution anomaly between climate change and the control baseline at the end of the century for all epipelagic communities. To understand the factors responsible of the distribution change, I compare the results with the sensitivity experiments. What I discovered is that the biomass decrease is mainly due to the decrease of plankton. The increased temperature, on the other hand, is the main factor controlling the distribution. Epipelagic communities tend to follow their thermal preferences, inducing expansion to the pole, what you can see here in red. About mesopelagic communities, the distribution is similar, but with almost no biomass increase in the northern hemisphere, except in the Gulf of Maine. As for epipelagic communities, their biomass decreases due to the decrease of plankton. The temperature is also responsible of their increased biomass in the Southern Ocean. But remember, they have no thermal preference, so this increase is due to their metabolic rate increasing. But the answer is not that simple. We have an indirect effect, which I will detail later. The vertical distribution also changes. In blue, we have the deepening of the organisms. This behavior is due to the deepening of plankton. As primary production declines, water clarity rises, letting light penetrate deeper. This shifts primary production, zooplankton, and their predators to greater depth. Mesopelagic residents are then controlled by different factors. As light penetrates deeper in the ocean layer, mesopelagic residents are forced to move deeper to maintain their preferred level of light. The oxygen minimum zone also influences mesopelagic residents locally, pushing them into shallower waters to avoid the core of the oxygen minimum zone, what we can see here in orange. Now, do you remember I told you we had indirect effect on mesopelagic communities? We understand this when we disentangle the effect of thermal preference and metabolism. So when we run the sensitivity experiment with only temperature under climate change, total biomass increases for everyone due to the expansion of the distribution and increased metabolism. Looking only at the thermal limits, the biomass increase only for epipelagic community. Mesopelagic should not change as they have no thermal limits. So why this plus 1% of biomass? It is because they benefit from epipelagic prey increases. When we look at the metabolism simulation, mesopelagic communities are clearly enhanced, exerting a predation pressure so high that epipelagic biomass decreases. In fact, the metabolism of epipelagic communities does increase their biomass when mesopelagic communities are not enhanced, as you can see here. So the increase of mesopelagic communities driven by the temperature factor is indirectly driven by epipelagic biomass increase thanks to their distribution expansion. I don't have time to develop the other sensitivity experiments, but I have, of course, looked at all factors and analyzed how they might interact to cause nonlinear effects. Climate-driven reorganization of pelagic ecosystems alter predator-prey interactions, forcing communities to adjust their diets regionally in response to changing prey availability. These dietary shifts are particularly pronounced within epipelagic communities. So today I will only present the small and medium epipelagic community. This community preys on a wide range of organisms, with mesozooplankton, macrozooplankton, and their own community forming the main part of its diet. Under climate change, zooplankton biomass decline and main predator size may change regionally. These structural changes in ecosystems lead to major trophic shifts, which result in regional changes in trophic fluxes of up to around 20%. In blue, the proportion of prey decreases in the trophic flux of small and medium epipelagic community, while in red, an increase in this proportion is observed. So what do we need to keep in mind? First, the responses to climate change are highly heterogeneous and vary according to each community's sensitivity to sometimes different forcings. It is therefore important not to look at biomass changes in aggregate terms. Then the sensitivity experiments make it possible to identify the key forcings for each community and to identify the role of each in the trophic chain. Spatial redistributions and variation in abundance will have a significant impact on trophic interactions, highlighting the resilience of certain communities compared to others. And last but not least, temperature has a direct effect on communities as well as indirect ones. To remind you of the issue addressed in the thesis, climate change increases pressures on marine ecosystems. However, pelagic ecosystems remain poorly understood, and few global marine ecosystem models are able to explicitly resolve the biological processes governing growth, metabolism, trophic interaction, and habitat use. Our study demonstrated that APECOSM accurately represents ecosystem structure and function of pelagic ecosystems. Pelagic ecosystems respond to a combination of physical and biogeochemical factors. Plankton and particulate organic matter emerge as critical supports for higher trophic levels, highlighting strong interdependencies between communities across depth layers. Under climate change, the results reveal pronounced community-specific responses, with biomass decline in tropical regions and poleward expansions. Sensitivity experiments show that primary production dominates global biomass change, while temperature primarily controls spatial redistribution. We saw that changes in biomass distribution and prey availability induce substantial shifts in predator diets, reflecting the adaptive capacity of pelagic communities. The mechanistic and trait-based modeling approaches contribute to a more process-based understanding of how marine ecosystems are vulnerable to climate change. They offer a foundation for future work aimed at improving projections of biodiversity, food web dynamics, and ecosystem services in a changing ocean. Despite all of the results, we have to consider their limits when interpreting them. Some regions are understudied, making it difficult to compare with our model outputs. The observational data are subject to uncertainties and biases, and the model itself has its own limitations. The coarse resolution does not allow to resolve fine-scale physical and biogeochemical features, while parametrization and processing representation are always under improvement. For example, mesopelagic resident response to oxygen is currently under adjustment due to some issue faced in the Chapter 3. They are also hugely dependent on particulate organic matter in the model, as zooplankton in Pisces do not go as deep as this community. Moreover, interpreting the response of mesopelagic feeding tuna community is often difficult as organisms' behavior changes through their life. In the last chapter, it would be possible to investigate multiple outputs further, such as growth and mortality rates, as well as the impact of El Niño throughout the century. In fact, I suspect that a strong El Niño followed by a 3 La Niña will increase nonlinearities for some communities. Therefore, it would be interesting to investigate this further. More generally, in this configuration, we worked only on pelagic ecosystems. However, pelagic communities also interact with benthic fish and demersal organisms that are not yet represented in the model. Parametrizing these communities in APECOSM will enable coastal ecosystems to be represented. In fact, these ecosystems interact strongly with human society and are of great socioeconomic importance. Moreover, this thesis examines the impact of climate change on marine ecosystems, but it doesn't address the role of fishing. Although climate change represents a major threat to ecosystems, particularly toward the end of the century, fishing remains nowadays the primary threat. To accurately capture the interplay between these two ecological pressures, fisheries must be incorporated into APECOSM. It is particularly important to assess the impact of policy-relevant fishery management strategy on food security and fishery livelihoods. Thank you for your attention. I just would like to take 2 more minutes for the acknowledgements. I would like to thank everyone who has crossed my path over the last 3 years, my supervisor, Olivier Maury and Mathieu Langaigne, and the engineer, Nicolas Barrier, who helped me so much at the beginning when I was completely lost. Thanks for sharing me your knowledge. I learned a lot from you, and I still have a lot to learn from you. A special thanks to Olivier, who proposed me this thesis when there was no reason to believe that I was capable of completing it. I would like also to thank my colleagues who are always kind and helpful. A special thanks to the young team and all non-permanent team who are very supportive and always up for a beach volley session or a meal. I would also like to thank my family. My parents weren't very supportive at the beginning, but huge thank for the support and the help at the end of my thesis. I would also like to thank Loic. Thank you for coming to see me so often in set. Thank for the cookies and the cakes, even if half of the time it's a fail. I would also like to thank my friends, especially my middle school friends, for these 19 years of support and love. And last but not least, I would like to thank my roommates who are very supportive and always there for me, especially when they are hungry. So thank you for your attention. I hope you enjoyed this presentation and that I managed to make it understandable for everyone. Thank you.